So which one are you guys on?
The last speaker for this session is Jingyi Jessica Li from UCLA, and she'll be telling us about quantitating translational control.
Thank you. Thank you very much for the introduction. And this is our recent paper about quantitating translational control and about decomposing the decomposing the translation rate into an mRNA abundance dependent and independent components. And we also investigate the mRNA sequences that specify each component. And I am an assistant professor from UCLA. So this is my joint work with Dr. Chu at Fred Hutchinson and Dr. Mark Bagan at Lawrence Berkeley Lab. And this is our paper on NAR last year. So the question we are interested in is to quantitate the central dogma, which describes how information flows from DNA to mRNA and then to protein. We know that there are 4 key steps that will determine the final protein abundance, including the mRNA expression, mRNA degradation, protein expression, and protein degradation. So we will consider all key steps in determining the protein variance. So this is a paper by Schwenhaus et al. In 2011 on Nature. So in this paper, they are the first study that did comprehensive genome-wide experiments to quantify mRNA expression, protein expression, and their degradation rates. So in their paper, they claim that translation is the most important step in determining protein abundance. And this is a figure from their paper, which find that the R squared between mRNA abundance and protein abundance is only 0.39, which is very low. However, in our study, we found that a main reason of having such a low R squared in their study is because they overestimated the variance of translation rates. So the black line here is the inferred translation rates from Schwenhaus et al. And the blue line is measured translation rates from ribosome profiling. So this is experimental data, and this is computationally predictive. So when we replace the translational rate variance using the measure variance, we obtained a different result. And this is what we got. This was reported in our 2014 paper and also in the Science Perspective article. So we found that it is very important to consider possible measurement errors before we make scientific conclusions. And motivated by the study, we continue our investigation in this area. And this is another paper from Causality et al. In 2015 on plus genetics. And this paper raised a very interesting point. That is the previous assumption that translation rates and mRNA expression are uncorrelated, or biologists think they are independent, may be questionable. So this is a toy example showing that in our previous hypothesis, we assume translation rates and mRNA abundance are uncorrelated. So therefore, if we look at protein abundance versus mRNA abundance, the scatter was explained by translation rate, but the slope should be approximately 1. So therefore, translation rate just add to the scatter. However, if translation rates are indeed correlated with mRNA abundance, and this was found by Causality in yeast, then we will see the slope between protein and mRNA should be greater than 1, not 1 anymore. So therefore, this motivates us to think, if we want to investigate the contribution of translation rates to protein abundance, we should consider 2 components separately, the part of the translation rate that are correlated with mRNA and the part that is uncorrelated with mRNA. And this is what we think. So this is a plot between translation rate and mRNA abundance. So we see that there's a positive correlation, and we decompose the translation rate into 2 parts. And because my collaborator as a biologist, they prefer to call it independent and undependent, so I just follow their terminology. So here, the independent part is just the residual, and the dependent part is what is linearly correlated with mRNA. So we do the decomposition, and after that, we can have the slope between the TR and mRNA from linear regression. And then theoretically, if our model that is here back to the central dogma, if the protein abundance is determined by MR expression plus translation, then on the log scale, then we should have the slope between protein and RNA as 1 plus the slope contributed by the translation rate. So this is our hypothesis. Then we can say with a good estimate of the slope, we can have a good estimate of the slope between protein and RNA. So we reanalyzed the Carsati et al. Data, and we found a very good confirmation of our hypothesis. So this is what we get from that approach. So we first estimated the slope between translation rate and mRNA abundance, and then we add 1. So using this approach, we get a slope about 1.22. We also did an independent approach by scaling the mRNA abundance according to some standards. So using this approach, and then we regress protein abundance on the corrected mRNA abundance, we get a slope of 1.17. And if you compare those two estimates and the confidence intervals, you can see that they agree pretty well. And we also want to make a note that our estimated slope between protein and mRNA is lower than the one in Carsati et al. Paper. The main reason is that they use the major axis regression instead of the least square regression. And we have argued that this is not a good choice in our paper. All right, so with this approach, you can see that after the decomposition of the translation rate into the mRNA independent and dependent component, we have this diagram to summarize the contribution. So because the dependent component is perfectly linearly correlated with mRNA, so its contribution is actually part of the mRNA contribution, and in terms of R square. And also its effect is to increase the slope between protein and mRNA from 1 to 1.20. The independent component contributes to the variance in protein, and that's this part. So with this, we can have a better understanding of how the different steps in the central dogma contribute to the variance in protein abundance. Then from a mechanistic perspective, we're interested in how some general sequence features contribute to translation rates in independent and dependent components. All right, so in our paper, we found 7 feature sets which are biologically meaningful and can well explain translation rates. So they include the 5' UTR length, the number of URFs, which are AUG and its variance in 5' UTR, the 5' UTR folding energy, and this is the element TICU we define in our paper, and I'll explain in detail. The length of the coding sequence and the amino acid frequencies in the coding sequence and the codons frequencies in the coding sequence. So they together explain 80% of the measured translation rate, which we think is very good. And this is our predicted translation rate, and this is the measured translation rate. One thing I want to make a note is that we just use a multiple linear model, and it has very good advantage of have a good interpretability. So that will allow us to explain the effects of individual features. All right, so the TICE, a definition in our paper, is called translation initiation control element. And it is an element we found that spans the AUG, the translation start site, and here in East, it's from negative 35 base pair to positive 28 base pair. The way we found it is from constructing a position weight matrix, and then found the length, the range, such that its PWM has the best explanatory power of the translation rate. So we found this region, and interestingly, this region has biological interpretation, as you will see in the next slide. And we also look at that the dinucleotide and trinucleotide frequencies in this region, and we found that including those frequencies, we can have better R square. So the biological interpretation of this element is if you look at the folding energy in this region, we look at the folding energy of the highly translated genes and the lowly translated genes. And you can see that this region corresponds to the biggest gap in the folding energy between the two groups. And also, since we have decomposed the translation rate into the mRNA-dependent and mRNA-independent components, we can investigate how much each feature contributes to each component. So here you can see from those bar plots that TICE explains better of the dependent component compared to the independent. Also, the coding sequence amino acid frequencies and codon frequencies. However, CDN's length is a different feature because it explains more in the independent component, and we will see each. So first of all, we want to provide explanation of why TICE and those amino acid and codon frequencies can explain better the dependent component. So this is an analysis we conducted because the codon frequencies is expected to be correlated with tRNA abundance because they are the materials to make amino acid, they are the materials to translate to amino acids. So you can see that we first see in the two components, if we look at their correspondingly high value genes versus low value genes, then there's a bigger gap in the R squared between the high genes and the low genes for the dependent part compared to the independent part. So this explains that the tRNA abundance is affecting more for the dependent part of the translation rate, which we think is very interesting. And this plot also shows the same message. Here we're showing the tRNA abundance here in the y-axis, and we're showing the ratio of the translation rate, the dependent part, between its top and bottom genes, and we're just taking the ratio for the codon frequency ratios. So you see that we expect there's a high linear correlation, and we only see this for the dependent part, but not for the independent part. So essentially, this result suggests that the tRNA abundance is only very informative for the genes whose translation depend on mRNA. And there's a paper by Presniak in 2015 actually agrees with our result. So they found that the codon usage increases mRNA stability. In other words, in their biological paper, they found that if there are a lot of ribosome on mRNAs, then that fast translation will increase the mRNA stability and make the mRNA also at a high expression. So this also suggests why we see there's a correlation between translation rate and mRNA abundance. And on the other hand, we see CDN's length, the feature is different because it explains more about the independent part. And why is that? So the biological explanation for this is because of this known phenomenon that CDN's length affects initiation rate. So we know that the ribosome will leave mRNA after it finishes translation, but due to the diffusion effect, it will be reattracted to the 5' cap and restart the translation again. So for short genes, because of this short length, the ribosome will be more easily attracted back to the 5' cap and reinitiate the translation. And this process has nothing to do with the number of copies of mRNA. So this agrees with our result that this feature explains better the independent component than the dependent component. So as a summary, we have found those features to explain translation rate, and they explain, some features explain better the component part, and some features explain better the independent part. And we think this provides better biological insights into the translation mechanism. And currently, our ongoing work is on other species. Our previous work is in the Cerevisidae East, and we're looking at other species listed here. And our goal is to find some general mechanism that is universal in other species. And I also want to make a note that here we are not studying the gene-specific features, such as microRNAs, and we're looking for the general mechanism. And again, this is our paper, and then I want to thank my collaborators here again. And also, I want to thank ISMB for selecting our paper as a talk, and also Johnson & Johnson for supporting this research. Thank you very much.
So, have you, this paper that you mentioned involving codon stability
Yeah.
makes me really curious about causality. Are you able to tease out the causality in your observation of this correlation between messenger RNA abundance and translational increase?
That's a very good question. I think if you want to discern the causality, I think the only way I think is to do some synthetic experiments, use synthetic biology. Like if you can actually have my ribosome on mRNA, dense ribosome on mRNA, and then study whether the mRNA become more stable or not.
And have you done
Yeah.
some of these experiments?
No, because we didn't do those experiments. Yeah, but that's, I think I agree that to understand which causes which is very important. Yeah.
Thank you.
Thank you.
So I have a couple questions. So the first one is, I was trying to understand the difference between the codon feature and then the one that says codon length.
Oh, the codon feature, this one is the codon frequencies. It's the 61 codons. So you know, like the trinucleotides that code amino acids.
Yeah.
That's the codon we're referring
So
to.
which
So
is the sixth one then?
that's right. It's the 61 features
No,
together.
that's the seventh, right? The seventh feature.
Yeah.
So the number 6 feature might be.
That's the amino acid frequency.
Oh, I
So
see.
we look at 20 amino acid frequencies. So you can expect that those two sets of features are highly redundant because the codons will determine amino acid. But here we just include them both to see whether we can have different interpretations of each. But actually the results are pretty consistent. The explanation is very similar.
Okay. So which one is ties?
Which one is what?
Ties.
TICE. So that one is what I explained here. So it's a combination of position weight matrices and dinucleotide and trinucleotide frequencies in this region.
Okay. All right.
Yeah.
Yeah. Thank you.
Thank you.
Let's thank Jingyi and all the speakers of the session for. Please don't forget to visit the post.